So, good morning, afternoon, or evening, depending on where you are. My name is Sanford. I'm a PhD candidate at the University of Alabama at Birmingham, and I will be discussing a phenomenon that's taken up the bulk of my dissertation process. So, one thing to bear in mind when you're thinking about mycoplasmas is that they're actually very well adapted to symbiosing. And one of the main ways that they've become so good at symbiosing or parasitism is they have a very dynamic surfaceome, so it's really hard to target. It's been very elegantly reviewed by Jonathan Arfi and colleagues down here. Really recommend this review article if you've never read it. But as you kind of see, a lot of the main ways this organism will prevent opsonization and other recognition from the host immune system is by having such a dynamic surface. Its proteome has a pretty low amino acyl tRNA fidelity. As you can see here, up to 1 in 20 amino acyl tRNAs will be mischarged in a mycoplasma compared to your typical like 1 in 3,000 range you'll see in most bacteria. There's a lot of proteolytic cleavage events that take place on the surface. As we've already kind of foreshadowed earlier on, a lot of cytoplasmic proteins will be shuttled to the surface where they will moonlight with a secondary function. Classic examples include like EFTU and enolases. You'll see in the next talk. It's also some great normal or typical factors such as phase variation, but we don't have to worry about those today. But one area that remains still pretty not well described with mycoplasmas is the glycome on the surfaces of these organisms, other than some of the capsular work that's been done over the past, I don't know, 80 or so years. But previous work from our lab has shown that some mycoplasmas, in this case, they use two murine pathogens as models, will glycosylate surface proteins. They do these by cleaving hexoses from oligosaccharides. So basically, as long as there's two or more units on it, a hexose can be cleaved off of it, and it does it in a pretty non-specific manner. On the proteolytic aspect of it, there's no amino acid consensus sequence like you'll see in a canonical glycosylation system. Initial identification so that serine, threonine, asparagine, and glutamine are able to be glycosylated. So that's a pretty wide range of glycosylation sites. And furthermore, once a hexose will get added to whatever amino acid has been hexosylated in this case, then the immediate area around that glycosylation site can't be glycosylated further, probably through some steric mechanism based on our current model, which I'll kind of outline in a little bit. We also have inclination to believe that this is an essential process. One of the main reasons being that we can't find a knockout anywhere in our transposon library. And as you'll see right here, we also did some screening of these minimal organisms. So my question then was for this project, is this conserved and minimal genome mycoplasmas? I'm using two organisms as models. I use Syn3A, which of course has an artificially reduced genome, which I contrast to M. genitalium, which of course has, I prefer to look at as a streamlined organism, which had evolved under the selective pressure of host immunity. So I think they offer intriguing foils to one another. I'm certainly not the first person to think this up by any stretch, but using these two together, we can get some pretty strong evidence that a process is essential for mycoplasmas, or at least quasi-essential in the case of Syn3A. But yeah, finding this process I thought would be a very exciting thing. So to screen for glycosylation, we use a parallel SDS page approach where we run equivalent lysate in two gels, stain one for protein content, in this case with Kumasi blue dye, and then stain for glycan content using ProQ Emerald 300. This is a periodic acid-based stain, which will help to interact with glycans that have been attached to proteins. And I did this in three organisms, actually. So we used the Mycoplasma mycoides GM12 genome, which of course is Syn3A's wild-type parent, Syn3A and MGen, as I already outlined. And upon seeing evidence of glycosylation in all three of these species, we excised two bands each from each species, as I outlined as A and B here, performed a trypsin digestion on them, chemically derivativized them, and then performed GCMS on those excised bands. When we did this, we noticed that it is predominantly, at least when these organisms are grown in SP4, because recall, this is more of like a scavenging system in the real world where a host oligosaccharide would be available to the cell. It would go, woohoo, take that, and popping on its surface protein. But in this case, since it's scavenging it from the sp4, it is predominantly glucose with a little bit of mannose, probably from the yeast extract and other products that you see there. So the next step then was how could we identify what sites are being glycosylated, kind of like we had shown at that slide a few minutes back there. To do this, I used a membrane enrichment process where we lyse the cells and then ultracentrifuge them down. If I wanted this to be a really definitive surface omic project, I would have probably done more of or try a different workflow with, say, biotinylation or something like that. But since really what I was more trying to do here is enrich the material, and I should say some of our previous data has pretty definitively shown that glycosylation is only taking place on the surface of these cells. And so with that in mind, once I have enriched for these surface proteins here by this ultracentrifugation process, we performed an in-solution trypsin digest along with lyse-C just to kind of increase our digestion efficiency. One thing, if you're ever working with the surface of these organisms, I really recommend using this detergent here called Waters Rapidgest. It really improved our digestion efficiency. Once this has been digested, we then used a Velos per Orbitrap and performed high resolution mass spectrometry, where we used these search parameters right here for variable PTMs. This is adopted from a M. genitalium proteomic workflow. Obviously, we added in hexosylation since that's the PTM we're interested in, but there was a pretty high degree of N-acetylation and an appreciable amount of oxidation of methionine residues. And also because Mycoplasma surfaces are kind of painful to work with, we also made sure to use our proteomics software to look for mutations using this algorithm called SPIDER and an unspecified PTM finder, which we added in larger glycans to try to search for those. So to kind of jump around a little bit for time limitations here, we only found hexoses being attached that passed our criteria here. This is a really intriguing bit of data that we found, or I'd say probably the coolest thing I have. This comes from EFTU. I'm calling this peptide A just for simplicity's sake, but here's the sequence right here. Predominantly within the population of this peptide, we have an unmodified peptide sequence. As you can see here by these mass shifts, we see both a threonine and a tyrosine residue here. However, within the population, we were also able to find instances where this threonine gets hexosylated, and then also different instances of that population where this tyrosine residue gets glycosylated. However, there's never one where both of them are getting glycosylated. So it helps to kind of support some of our variability hypothesis. This is kind of like a white whale that I was searching for a long time, an instance of modification within a single peptide, or numerous instances of modification. And in fact, when you alpha fold the SYN3A EFTU structure, which is a pretty well conserved structure to other bacterial EFTUs, we were able to see that both of these potential glycosylation sites do seem to be available to the solvent or exposed to the solvent. And as outlined here, kind of increased our confidence that we didn't get a computer false positive there, along with our pretty robust statistics I forgot to mention here, but these are all well below regular p-values of 0.01. The other really noteworthy thing about this, or the other bit of data that really shocked us, is we expanded out our glycosylation pool or site pool to include the acidic amino acids. And we were actually able to find within this fatty acid binding protein that aspartic acid is able to be glycosylated here, which to our knowledge is the first time that's ever actually been reported in the literature. I think this is probably a phenomenon. This phenomenon is probably mediated by kind of how I showed how sloppy their metabolisms tend to be, or non-specific their enzymes might be another way to put that. So taken with that data in mind, we think, or we're willing to conclude that this glycosylation system is conserved within these three organisms, particularly SYN3 and MGen gives it a really strong evidence of essentiality in tandem with that inability to find a knockout, as we mentioned. And we also see that there is a wider range of acceptors than we had previously expected there to be. And we're quite shocked by that. So this is basically kind of an outline of how we think this mechanism is taking place here, where some unknown enzyme is mediating the cleavage of this precursor oligosaccharide and then attaching it to surface proteins without a motif to it. So very briefly, the next thing that we want to do is kind of one thing we'd like to try to see if we can find what genes are responsible for this phenomenon. I'm trying to get my hands on some synth 3.0 and compare it as soon as I can. If it's not in synth 3.0, then I could reasonably deduce it's within the addition of those 19 genes into synth 3A. And if it's still present in synth 3.0, then it's going to probably be more of like the scope of a postdoc project where you'd have to do a lot of in vitro assays if you wanted to find this. One thing that's more simple that we're trying to do is find out what initial conditions will affect the system. Like, basically, if we're feeding it different precursor glycans, do we see differences in glycosylation? So, for example, like does the linkage or ring position of the glycosidic bond count? Or does what particular moiety being the reducing sugar, say a galactose versus a glucose versus a mannose or what have you, affects this phenomenon? So with that, I'd just like to thank my lab mates and collaborators, and thank you for your attention.
Really nice, John. I was curious. I wish I knew you showed a proteomics, I mean a GenBank accession for the protein. I'm curious if you knew what its locus tag was.
Offhand, I don't know the locus tag. I got the GenPeps accession here, and I do know it is annotated as a fatty acid binding protein, but offhand, I can't recall right now what the locus is.
I will look about that on my own. So, as I remember, with wild type Mycoplasma mycoides, you do get the disaccharides binding, right?
Mm-hmm.
Yes, I'm sorry.
Yes. Oh, yes.
Okay. Sorry. And so, any idea what we lost to make that happen? Does that my question make sense? I mean, what gene enables disaccharide binding, but you can't do that with SYN1?
Oh, I kind of arbitrarily chose GM12 versus SYNth1. I just kind of had more easy access to that when I was writing this.
Sure.
Sorry, I might be misunderstanding because I believe that we have evidence of the disaccharides being incorporated in both organisms.
Well, I thought you said you only got monosaccharides incorporated in SYN3A.
Oh, I'm sorry. So to my based on what we understand about this process, it seems like we don't ever see disaccharides or larger being incorporated into these glycoproteins. Sorry for my misunderstanding there.
Okay.
And that's including in the wild type and any other instance that we've seen. I've never been convinced, I should say, by any of the disaccharides or larger.
But with just glycosylation, will that mean that the proteins will stay cytoplasmic?
So this, we believe, is happening on the surface. We've done staining when we, lyse cells. I believe this was in the murine mycoplasmas, at least, though. But when we try to stain cytoplasmic fractions, we get no signal that's discernible from the background. So we think it's specifically happening on the surface of these organisms.
As in a process that is on the outside. So
that's what we believe, not like inside and then flipping out.
So this would be proteins that sort of roam the surface of the cell and bump into these. I mean, so the proteins have to be in the membrane for them to get glycosylated.
That's what we believe, yes.
Okay, but they don't have to have transmembrane regions for that to happen?
No, we don't think so. As long as it's solvent exposed, we believe that's more of what is governing the availability of the amino acid to be glycosylated. So instead of when you'd see a conventional sequence consensus motif for a UDP a UDP sugar to get attached to it, We think this one is, yeah, basically it's just an availability of any protein that's on the surface of an amino acid that's amenable to being or having a hexose attached to it. Once that exposed solvent, or say the solvent exposed glycosyl receptor is sitting there, then they can get attached basically. So we think it's just more like a stochastic or maybe a chaotic process. So it's definitely going to make the modeler's problem or life's a bit harder if they want to put this in.
And so how do proteins without the transmembrane regions make it to the surface of the cell?
I suppose that's a good lead in for what James is talking about. But yeah, so there's a phospho sugar basically that will, at least in the other mycoplasmas, I should say a rhamnose phosphate will be used as a kind of a signal for the, I should say the cytoplasmic protein to be sent off to the surface and then be linked that way to the lipid moiety.
But I thought that Centria doesn't do rhamnose.
Yeah, so I was going to say that's the next question too. I say so. Typically, we think it's rhamnose. In this case, they might be a different glycan because rhamnose synthesis has been knocked out.
Mm-hmm.
Sorry, I'm like stealing James's thunder right now.
Okay, I'll stop.
You're fine. Yeah, no problem.
Other questions for John?
Yeah, this is Kim Wives. A couple of real quick ones, John. I think that the protein that you mentioned about the fatty acid binding protein, I think that's by modeling other organisms, that's supposed to be anchored in the inner leaflet of the membrane, so it might be an internal. And number two is a question, do you have any evidence that rules out the fact that the glycosylating enzymes come from the medium rather than the mycoplasma, that are not encoded, but rather they're from the environment?
Right. So one of the ways that I can increase my confidence for that is there's always like a background of like serum proteins within the medium. And well, normally like, our proteomics software will filter those out as contaminants. I do take a look at those and I haven't seen, or when I see instances of hexosylation of those serum proteins that aren't actually within the mycoplasma surface, those ones will not have hexoses in these kind of more novel attachment sites, and also happens at a pretty low abundance anyways. So I'd say that's my strongest evidence to counter that.
Okay, thanks.
Well, thank you.
Okay.
Good question.